Okay. So I am recording now. So like I told you before, I am going to post the recorded lectures on eCampus. So I had problems with Zoom Tuesday, but this time, finally now, everything seems to be working. We're recording, and this will be posted on eCampus for you guys to refer to later if you need to. Okay, so nucleic acids. Oh, great, another technical difficulty. My remote is out of batteries. Oh, no, it isn't. Okay. Okay. So again, so here's some historical context. So we're going to kind of follow the timeline of history, just like we did with Mendelian genetics. So from about the 1900s to the 1300s, there's a couple things we knew about not just genetics, but about the molecule that passes on genetic information. So let's go back to Mendel for a second. So Mendel did his work at the monastery around 1863 or so, okay, around the time of the American Civil War. One thing I failed to mention on Tuesday was you would think that a huge discovery would make a big splash, right? This would be a very famous paper, and it would be reported on. But actually, when Mendel published his discoveries, it didn't make a splash at all. Okay, nobody really noticed. And in fact, his paper was completely forgotten about until the turn of the century. Right around 1900, three German botanists reproduced Mendel's results independently. Okay, and then when they did a literature search, published their findings, they find that actually the results were already discovered and published by an Austrian monk years before. Okay, so Mendel's results are completely forgotten about. Turn of the century, these three German botanists rediscover Mendel's work. And then right around that time, 1901, is when Sutton is studying meiosis, and he sees that one sister chromatid from a pair of sister chromatids goes into gametes, and that is entirely consistent with Mendel's first law. Okay, so it confirms Mendel's work. Okay, a little bit later, we have Thomas Hunt Morgan. So from Sutton, we know that these chromosomes, chromosomes have something to do with inheritance because they're consistent with Mendel's laws. Okay. Then Thomas Hunt Morgan, by using recombinant phenotypes to map the positions of genes on a chromosome, he established that the genes are actually contained on the chromosomes. All right, but at this point, scientists still don't know which molecule actually carries the genetic information. So remember that chromosomes are made from chromatin. A chromatin is a mixture of DNA and proteins, okay, specifically scaffolding proteins, histones, that help to condense the chromosomes down into the sister chromatids. So there was a debate at this point. A lot of people said, well, proteins are the molecules, obviously, that carry genetic information. Other people thought, well, no, DNA, nucleic acids, are the molecules that carry genetic information. Okay, and in fact, the dominant theory was that proteins were the carriers of genetic information, and that nucleic acids were simply there as scaffolds in order for the structural integrity of the chromosome. Okay. Nowadays, actually, we know it's exactly the opposite, that DNA carries genetic information, and the protein is there as a scaffold, for the structural integrity of the chromosomes. But that wasn't the predominant that wasn't the predominant hypothesis at the time. Okay, so So slowly over the course of a couple classic experiments, evidence started to accumulate that DNA was actually the molecule that carried genetic information. Okay, so first, let me turn away. So Griffith's experiment, this is a classic experiment. And while it didn't really indicate that DNA was the molecule that carried genetic information, it did identify something called the transforming principle, which is really important. So in his famous experiment in 1928, Frederick Griffith showed that a transformation principle in a heat-killed virulent strain of bacteria can confer virulence to a living non-virulent strain. Okay, so pay attention to this figure down below. And I'll explain exactly how Griffith's experiment worked. So Griffith was working with two strains of Streptococcus pneumoniae. One called the S strain, which was virulent. In other words, it was able to cause disease. And one called the R strain, which would not cause disease. Okay? S and R in this case, it stood for smooth and rough. So when Griffith cultured these two strains, the one strain, the S strain, appeared smooth. The colonies appeared smooth, whereas the colonies of the non-virulent strain appeared to be rough. The reason for this is the S strain is an encapsulated strain. So some strains of S. pneumoniae express polysaccharide capsules. Others do not express polysaccharide capsules. And it's the strain that expressed the polysaccharide capsule that was virulent, able to cause disease. Okay? So he did this series of experiments. So he did these experiments in a mouse model. So he cultured the S strain. This is the smooth strain, which is virulent. Injected that into a healthy mouse, and the mouse dies. It grew up the rough strain, the non-virulent strain, unencapsulated. It injects that into another mouse. The mouse remains healthy. Then he took some of the S cells and killed them by heating them up. Okay? He injected the killed S strain into a mouse. The mouse remained healthy because the cells were heat killed. And this is the interesting experiment. He took the heat killed virulent S strain, mixed it with living R strain, and then injected it into another mouse. The mouse died, and then S cells were cultured from the dead mouse. Okay, so dead S cells, live R cells were injected. We know that the heat-killed S cells don't kill the mouse. Okay, we know that the living R strain doesn't kill the mouse. Mouse dies, and we recover living S strain. Okay, so from this, Griffith hypothesized that there's a transforming principle, a transformation principle. Okay, so he knows that there's something in the heat-killed S strain that transformed the R strain into a virulent strain. Okay. Now again, at this point, that transformation principle hasn't been identified as DNA yet, but we know that there's something there. Okay, so that was in 1928. About 16 years later, 1944, Oswald Avery repeats Griffith's experiment, but he does something different in this case. Okay, so Oswald Avery shows that adding the oxyribonuclease stops transformation, while adding a protease does not. For the first time, DNA is implicated as the medium of the genetic information. Okay, so if we consider this last experiment over here on the far right-hand side. What Avery did is he added, in two different experiments, he added enzymes. Okay, a deoxyribonuclease is an enzyme that breaks down DNA. A protease is an enzyme that breaks down proteins. Okay, so if the transformation principle is DNA, then adding a protease shouldn't affect it. If it's protein, adding a protease should ablate. It should get rid of that transformation principle. Okay, same thing for DNA. If DNA is a transformation principle, then adding a deoxyribonuclease should break down the DNA, and we shouldn't see transformation. However, and a protease shouldn't affect it. The results of the experiment were that adding a deoxyribonuclease prevented transformation of the R strain into the S strain, whereas adding a protease didn't affect it at all. Okay, so now finally, for the first time, DNA is implicated as the molecule that carries genetic information. Okay, so a couple of years later, we have another experiment that also adds some more evidence that DNA is the molecule that carries genetic information. Again, this is another classic experiment. This is the Hershey and Chase experiment. So this is Alfred Hershey and Martha Chase over here. And they used radioactively labeled bacteriophage in order to carry out their experiment. Okay, and I won't draw on the board just yet. Okay, so here's the Hershey and Chase experiment. So those of you in class, how many of you have had, I guess all of you have had micro already, right? And have you studied bacteriophage? You went over bacteriophage, right? For those of you joining by Zoom, okay, a bacteriophage, for anybody who doesn't know, is a virus. Okay, and it's a virus that infects bacteria. So, and really, they're my favorite of all the viruses. They're really cool looking. And there was a lot of really interesting research done on bacteriophage as well. So if you're looking at bacteriophage, it has an icosahedral head that looks like this. Okay, the icosahedral head, this is where the DNA is. Okay, then it has a neck that looks like this. It has legs that kind of make it look like a lunar lander or a spaceship. And when it attaches to the bacteria, it actually injects the DNA through the neck into the bacteria, much like a hypodermic syringe injects vaccines into your arm. Okay, so that's a bacteriophage, really cool little viruses. Okay, so let's look at exactly what happened in the Hershey and Chase experiment. So again, Hershey and Chase, just like Avery Oswald did, is looking at whether it's protein or DNA that carries genetic information. So in the first experiment, they're using bacteriophage whose proteins are radioactively labeled. Okay. So they have phage that are radioactively labeled. They mix them together with bacteria, and of course the phage are going to infect the bacteria, right? After letting the phage infect the bacteria for a certain amount of time, they were put into a plain old Waring blender. Okay, the reason for the blender was they blended for a short amount of time. This is to detach the phage from the surface of the bacteria. Okay. Then they took the bacteria and they centrifuged it and tested for radioactivity. Okay, now bacteria being more dense are going to be located in the pellet. Okay, they're going to be centrifuged down to the bottom of the pellet. Okay, the phage themselves are less dense, so they're going to be in the supernatant. So when they tested for radioactivity, they found that all of the radioactivity was in the supernatant rather than in the pellet. Okay, so this disqualifies protein as the medium for carrying genetic information, because if it was carrying genetic information, it would co-locate with the bacteria. It would be in the bacteria. Okay. Then they did a different experiment. In this experiment, they radioactively labeled the DNA of bacteriophage. Again, they mixed them with bacteria, gave a certain amount of time for the phage to infect bacteria. Okay, then again, they put them in the blender. So the blender is going to detach the phage from the surface of the bacteria. Okay, then again, they centrifuged. Okay, again, so the centrifuge is going to, so the bacteria are going to be in the pellet, and then everything that's all the soluble matter is going to be in the supernatant. Okay, this time, the radioactivity co-located to the pellet. So this means that the radioactively labeled DNA from the phage is in the bacteria because the radioactivity co-located to the pellet. Okay. So again, this is another line of evidence that DNA is the molecule that carries genetic information. Okay, so by this time, so now we're at 1949, so we're getting about the mid-20th century here. In 1949, Erwin Chargaff looks at the relative amounts of the 4 DNA bases in different species. He finds that while the exact ratios of nucleotides varies from species to species, the amount of A always equals T and the amount of G always equals C. Okay, so at this point in time, there's been a couple experiments that indicate that DNA is actually the carrier of genetic information. However, we don't really know too much about the structure of the DNA molecule yet. Okay? That's going to come in a couple of years in 1956 when Watson and Crick do their famous experiments to show that it's the double helix. Okay? At this time when Chargaff is doing his experiments, they still don't know the structure of DNA. Okay, however, he found something really interesting. So we're looking at some different species, Homo sapiens, humans, of course, Drosophila, Melanogaster, these are fruit flies, Zea mays, corn, Neospora crassa, this is a fungus, Escherichia coli, E. coli, of course, is a bacterium, Bacillus subtilis is a bacterium as well. Okay, and we can see there's different amounts of the 4 bases, but if we look at just 2 bases, so let's look at Homo sapiens, A and T, 31.5%, G and C, 19.1, 18.4, about the same. Fruit flies, Drosophila, 27.3, 27.6, so about the same amount of A and T, exactly the same amount of G and C. And as we go through all the different species, although the numbers are different, the amount of A and T is always the same, the amount of G and C is always the same. Okay, so let me ask you guys, and I'll walk over here so I can see the chat from Zoom. Why is this? What do we know about the structure of DNA now that explains Chargaff's results? Yeah, what do we call that? Base pairing. Base pairing, exactly. So of course now, years later, after Watson and Crick have discovered the structure, we know that A is always base, we have DNA is a double helix, A is always base pair with T's, C is always base pair with G's, which explains Chargaff's results precisely. All right, 1953, I said 1956, I was a little bit late. So 1953, James Watson and Francis Crick proposed the complementary double helix model of DNA based on the X-ray diffraction data of Maurice Wilkins and Rosalind Franklin. So what we're looking at here, this is a very, very famous photograph that you've probably seen before. This is called photograph 51. And what this is a photograph of, this is X-ray crystallography. Rosalind Franklin was really an expert X-ray crystallographer, and she's the one who did the work that and generated this picture. So in case you don't know, what X-ray crystallography is you isolate a whole bunch of a molecule, and then you crystallize it. Crystallizing is really the hardest part of X-ray crystallography. Okay, so you crystallize it so that it forms a crystal with a regular structure. Then you shoot an X-ray, a beam of X-rays at that crystal, and you have a piece of photographic film behind it. And when the X-rays hit the crystal, depending on the structure of the crystal, it's going to diffract the X-rays in a certain pattern, and then that pattern will be recorded on the film. So wherever the diffracted X-rays hit the photographic film, it's going to expose it and turn it black. And based on the patterns that we see on the photographic film, we can determine the structure of the molecule in the crystal. Okay. What we're looking at here, the important thing about photograph 51 is this X. Okay. Whenever you see an X in X-ray crystallography, that means double helix. Okay, so this was really the light bulb moment. When we looked at photograph 51, boom, double helix. And the rest of the pieces started to fall in place after photograph 51 identified a double helix in the structure of DNA. Okay, so let's talk about the structure of DNA. So obviously we understand a lot more about the structure of DNA now. Strands, so DNA is a double helix composed of two strands of DNA. So it's a double-stranded helix. The strands are held together by hydrogen bonds, specific AT and CG base pairing. So the bases are in the center of the helix, held together by hydrogen bonds. Okay. A's always base pair with T's, G's always base pair with C's. Again, this explains Chargaff's results. These are Chargaff's rules. Each strand has a 5' to 3', pardon me, a 5' to 3' polarity and runs anti-parallel to the complementary strand. Okay, so let me draw that on the board. That black didn't show up very well. Do I have an eraser? I do not have an eraser. Does it show up better? Yeah, that red shows up a little bit better on the camera. Okay, so DNA, just like proteins, have a directionality or a polarity. If we look at nucleotides, so let me draw a very simple nucleotide. So, like I mentioned, no, I'm sorry, I mentioned that in my other class. DNA is a polymer, it's a biopolymer. Okay, and polymers are made up of individual monomers that are strung together into a polymer. The monomers that make up DNA are molecules called nucleotides. If we look at the structure of a nucleotide contains a 5-carbon sugar. It contains a nitrogen-containing base. This is either A, T, C, or G. And it contains between 1 and 3 phosphate groups. Okay? And the carbons on this 5-carbon sugar, we number them 1', 2', 3', 4', and 5'. Okay, so over here is the 1' carbon, 2', 3' here has a hydroxyl, 4' is over here, and 5' is over here outside of the ring. Okay. Now, when we join nucleotides together in order to make a nucleic acid, we join the 5' phosphates of one nucleotide to the 3' hydroxyl of another. Okay, so that means that DNA is always going to have a 3' end where we have a free hydroxyl and a 5' end where we have one or more free phosphate groups. Okay, so this gives a single strand of DNA a polarity or a directionality. Okay, and we always talk about this in the 5' to 3' direction. Okay, so we have a 5 to 3 polarity. So that's a single strand of DNA with a 5' to 3' polarity. And like I mentioned, the strands that make up a double-stranded helix, the double helix of DNA, have 2 anti-parallel strands. Okay, so we have one strand 5' to 3' in one direction, and the other one 5' to 3' in the opposite direction. Okay, so double-stranded anti-parallel with respect to their polarity or their directionality. Okay, let's go back to the PowerPoint. Okay, so we can see that here. Look at the figure. So here's a molecule of DNA. Okay, again, double-stranded. So we have one strand here, another strand here, twisted into the helix. In the center is where we have the bases. Okay, those bases are going to base pair and be held together by hydrogen bonds in the center. And again, anti-parallel. So here's the 5' end of one. Okay, so here's the 5' end. We follow that down 5 to 3, and the other strand is in the opposite. So we have 5' here, 3' here. Okay. Also notice the genetic information is in the center of the helix. Okay, the sequence of base pairs is where the genetic information is contained. The structural part of the DNA molecule is what's called the phosphosugar backbone. Okay, so the phosphosugar backbone, we can see here's the phosphosugar backbone of one, and here's the phosphosugar backbone of another here. Okay, that's the structural portion. That's what holds the whole thing together. Okay, so once Watson and Crick finally solved the structure of DNA, it explained an awful lot. One thing that it explained was how DNA could replicate itself. The double-stranded helix model explains this perfectly because each strand contains all of the information that you need to synthesize a new strand. Okay? So this structure conceptually explains the manner in which genes replicate. So when we split the DNA molecule in two, each single strand can act as a template that contains all the information for synthesis of a new complementary strand. Okay. It's actually a very elegant sort of solution to that problem. Okay, so now we're in 1956. Kornberg develops a system of in vitro replication and discovers that DNA building blocks are high energy precursors, DATP, DTTP, DGTP, and DCTP. A single polypeptide DNA polymerase 1 catalyzes the synthesis of new DNA strands. Okay. So first of all, it says Kornberg develops a system of in vitro replication. What exactly does that mean? What is in vitro replication? How is it different than how replication occurs in organisms? Anybody know? Anybody from Zoom want to chime in on chat? Okay, the word in vitro, it comes from Latin. In vitro literally means in glass. Okay, so in vitro literally means in glass rather. So in vitro replication means replication, a system of replication that can be done in a test tube, okay, free of cells. So this was actually a really significant development. So he was able to, just by adding different enzymes, different precursors into a test tube, he was able to get replication of DNA. Okay, so he discovers that the building blocks of DNA are high energy precursors, the ATP, DTTP, DGTP, and DCTP. Okay, these are all nucleotides. Okay, so A, T, C, or G, and the little letter D in front. This indicates that they're deoxy-GTP, deoxy-CTP, deoxy-ATP, and deoxy-TTP. The difference is presence or absence of a 2' hydroxyl. I think this is something that we'll cover in more detail later. But DNA, the 5-carbon sugar that makes up DNA is deoxyribose. Okay, deoxyribose lacks a 2' hydroxyl. The nucleotides that make up RNA have ribose as the 5-carbon sugar. Okay, and ribose has a 2' hydroxyl. Okay, so this is an RNTP or a ribo. Rather, this would just be an NTP or a nucleotide triphosphate. When we have deoxyribose as a 5-carbon sugar, that's a DNTP or a deoxynucleoside triphosphate. Okay. Also, a single polypeptide DNA polymerase 1 catalyzes the synthesis of new DNA strands. So also he discovered there's actually many different DNA polymerases, but Kornberg also discovered the enzyme DNA polymerase 1. Okay, the nucleotide precursors are linked 3' to 5'. So like I showed you before on the board, the 3' hydroxyl of one nucleotide is linked to the 5' phosphate of another nucleotide. Okay, so that gives us the 5' to 3' polarity. Polymerization only works in the presence of template of DNA or template DNA. So this is another important discovery from Kornberg's experiment. Usually you give an enzyme precursors, and you put in the enzyme, you put in the reactants, and you get the product. It didn't work that way with DNA polymerase. You had to put in the reactants, you had to put in the enzyme, and you also needed DNA to act as a template. Okay, so this indicates that the information for synthesizing a new strand is contained on another strand. So you need another strand to act as a template in order to make more DNA. The new strands had the same ratio of AT to GC base pairs as the template DNA. Okay, so again, when you make a complementary strand, we're going to have base pairs, right? A's are going to base pair with T's, C's are going to base pair with G's. So that's going to give you the same ratios as you had in the template strand. Okay, so Kornberg discovered some important things. He discovered, that this is all done by a single enzyme called DNA polymerase. He discovered that you need DNA to make more DNA, so he discovered that you needed template DNA. He discovered what the precursors were, the DNTPs. However, what he didn't know yet is exactly how DNA replication takes place. So there were 3 different models out there at the time describing how DNA replicates. First, at the top, there was the conservative model. So in the conservative model, this hypothesized that a brand new double-stranded DNA molecule is synthesized from the information on the existing DNA. Okay? So in other words, so here's the conservative model over here. So here's the template strands of DNA. So according to the conservative model, the template strands of DNA will be used to make a brand new double-stranded DNA molecule, which is over here. Okay, so we retain the same old template strands, and we get the brand new, brand new double-stranded DNA molecule, which is in blue. Next, we have the semi-conservative model. In the semi-conservative model, the 2 strands of the template are distributed between the daughter strands. So in the semi-conservative model, the double-stranded DNA is denatured into 2 single strands, and each single strand is used to synthesize a new daughter strand. So what we get then are 2 hybrid DNA molecules, okay, composed of 1 template strand here and 1 newly synthesized daughter strand in blue. Okay. Finally, the last model was the dispersive model. In the dispersive model, the strand is broken up frequently, and the broken parts are used as primers to prime synthesis of new DNA. So what we would get in the dispersive model is a mixture of old DNA and newly synthesized DNA all throughout the 2 new molecules. Okay, so we have these 3 models for DNA replication. So next we have to figure out exactly which model is accurate. Okay. So this is another one of the classic experiments that I want to explain to you. In 1958, Nielsen and Stahl used DNA isolated from bacteria grown in a heavy isotope of nitrogen and density gradient centrifugation to determine which model of DNA replication is true. Okay, let me explain exactly how we do the experiment. Then we'll look at the results. Tell you what, why don't we use this other part? All right, I'll use this part of the whiteboard. Okay, so the first thing that Muses and Stahl did was they grew up bacteria in medium that contained this heavy isotope of nitrogen, nitrogen 15. Okay, so here's a bacterial culture. We'll just put this in a Erlenmeyer flask. And this is bacteria grown in N15. Okay, so they have this growth medium. It's labeled with heavy nitrogen. They put their bacterial culture in, put it in the incubator overnight. Okay, come back next morning. All of the bacteria grown overnight have heavy nitrogen incorporated into the DNA. Okay. The next morning, they take the bacteria, centrifuge them, wash them, and put them in normal medium that doesn't have N15. Okay. So in the morning, they take out the bacteria, centrifuge the bacteria, wash the bacteria so they get rid of that medium that contains the heavy nitrogen, and then they switch it to normal medium. You guys all took microbiology, so this would be like normal LD broth, or I forget exactly what Dr. Farrar and Dr. Williams used, but this is just normal growth medium. All right. Now, we know that the generation time for bacteria, specifically E. coli, the generation time is 20 minutes. So every 20 minutes, we're going to get a new generation of bacteria. The bacteria are going to help. So what Muses and Stahl did next is they took samples every 20 minutes. So the generation time of E. coli is 20 minutes. So after 20 minutes, we should have one round of replication of DNA. Okay, after 40 minutes, we should have 2 rounds of replication of DNA. Okay? So let's just stop at 2 rounds of replication of DNA and look back at the results of the experiments on the PowerPoint. Okay, so these are the results that we would expect for each of the 3 models that were proposed. Okay, so first for conservative replication. So at the 0 time point, okay, after the bacteria had been grown in heavy nitrogen medium overnight, we would expect just 1 band. Okay, first let me explain what they're doing here. This is density gradient centrifugation. Let's go back to the board. So the samples that Jesus and Stahl took, they analyzed them by density gradient centrifugation. So density gradient centrifugation, what you do is you take a test tube, and this test tube has a concentration gradient. Okay, when we're analyzing DNA, we use a cesium chloride concentration gradient. Okay, so the concentration of cesium chloride, it's very high here, and it gets less and less and less, and the lowest concentration of cesium chloride is here. So we have the cesium chloride concentration gradient. When we apply the DNA samples to the top of the cesium chloride gradient, they're going to migrate down until they reach an area of the gradient that's the same density that they are. Okay. N15 is a heavy isotope of nitrogen. So N15 labeled DNA is going to migrate farther down the cesium chloride gradient than unlabeled DNA. The unlabeled DNA is lighter, so it's not going to travel down the cesium chloride gradient as far. Okay, so this would be N15 labeled DNA farther down the cesium chloride gradient. Unlabeled DNA won't go as far down the cesium chloride gradient. Okay, now let's go back and look at the results of the experiments. Okay, so let's look at the results that we would expect for conservative replication. Okay, so in conservative replication at the 0 time point, we're just going to see one band of heavy DNA labeled with N15, so it's going to go farther down the cesium chloride gradient. After one round of replication, we're still going to have the original DNA that acted as template DNA, but we're going to have another band of DNA made all of light DNA. Okay, because remember with the conservative model, we're going to use the template DNA to make a brand new double-stranded DNA molecule. Okay, after 2 rounds of replication, would expect the results to be the same. Okay. With the dispersive model, okay, again at the 0 time point, we're going to have one band of heavy DNA. Because with the dispersive model, the new DNA is going to be all throughout the new DNA molecules, kind of all over the place. So this means as we go through subsequent rounds of replication, that DNA is going to get lighter and lighter. It's going to the DNA, the heavy DNA is going to be kind of titrated out, and the band's going to get higher and higher. Okay, so after one round of replication, we're going to see the heavy band is going to disappear because we're going to be adding new DNA to that template DNA. And after another round, it's going to get even lighter, and it will continue to get lighter and lighter and lighter with subsequent rounds of replication. Okay, now with the semi-conservative model, again, we're going to start with one band of heavy DNA. After one round of replication, we're going to have hybrid DNA molecules. Okay, made of one labeled heavy strand that acts as a template and one newly synthesized strand. So we're going to have one labeled strand, one unlabeled strand. After another round of replication, we're still going to have one labeled strand, one unlabeled strand, but we're going to have more unlabeled DNA as well. Okay, so after one round, we expect a band of hybrid DNA molecules. After another round, we're still going to have some hybrid molecules, and we're going to have some DNA molecules that are entirely made out of the light unlabeled DNA. Okay. Now, when Michelson and Stahl analyzed the results of their experiment, the results were here. Okay, and obviously those results are exactly the same, exactly what we would expect for the semi-conservative model of replication. Okay, so Michelson and Stahl's experiment confirmed the semi-conservative model of DNA replication. Okay, is everybody good on that? Okay, it's kind of a complicated experiment to understand, but it's a very important experiment. So I do want you guys to understand the Michelson and Stahl experiment. Okay, so now we've solved these. So we know at this point in history, we know that DNA is the molecule that carries genetic information. We know the structure of the DNA molecule, and we know how the DNA molecule replicates itself. It replicates itself by semi-conservative replication. However, even back at this point in time, there's a problem. Even at this point in time, we knew that DNA never leaves the nucleus. It's always in the nucleus of the cell. And even back at this point in time, we knew that synthesis of proteins took place in the cytoplasm. Okay, so the question now is, how does the information contained in the nucleus of the cell get out to the cytoplasm where proteins are being made? Okay, so even back in the 1950s, it was known that in eukaryotic organisms, DNA is located in the nucleus, while protein synthesis takes place in the cytoplasm, where DNA is absent. So the DNA double helix cannot be the template for protein synthesis. Therefore, there must be a second molecule that gets its specificity from DNA, but is transported to the cytoplasm to act as a template for protein synthesis. Okay, so there has to be, they knew there had to be some intermediate between the DNA and where protein synthesis takes place in the cytoplasm. Okay, so one candidate for that intermediate is RNA. So even back then, we knew that RNA is found all throughout the cell. Okay? It isn't just in the nucleus the way that DNA is. Okay. Structure of DNA is similar to structure of RNA rather is similar to DNA in certain respects, dissimilar in others. Similarities: RNA, like DNA, is a long unbranched molecule made of 4 different types of nucleotides. Okay, nucleotides are a little bit different. So in DNA we have A's, T's, G's, and C's. In RNA, we have A's, U's, G's, and C's. Okay, U standing for uracil. Also, similarly, these nucleotides are joined 3' to 5', so we still have that 3' to 5' polarity. Okay, the individual monomers are still the 5' phosphate of one monomer is still joined to the 3' hydroxyl of another one to make a long linear polymer. Unlike DNA, RNA does not contain, so these are some of the differences between DNA and RNA. Unlike DNA, RNA does not contain thymine, but rather the closely related pyrimidine uracil. Unlike DNA, RNA contains the sugar ribose rather than the sugar deoxyribose. Okay, and that's what I was showing you here before. Okay, so going back to my drawing. See if you can see this better if I zoom in a little more. Oh, there we go. All right, and this bar is right. Okay, sorry for those of you at home, the bar there is kind of blocking my nucleotide right at my 5 carbon sugar. So one of the differences between DNA and RNA, the nucleotides that make up DNA have deoxyribose as the 5-carbon sugar. Okay, this is deoxyribose. The deoxy part of deoxyribose refers to the fact that it lacks a 2' hydroxyl. RNA has ribose rather than deoxyribose. So if I draw in a 2' hydroxyl here, okay, that's deoxyribose. So that makes this, and actually if we change this from a T to a U, okay, now we have a nucleotide that makes up RNA rather than a nucleotide that makes up DNA. Okay, and it looks like we're about out of time, so why don't we stop here? We'll continue with this PowerPoint on Tuesday. Also remember this coming Tuesday, we are going to have a lab. So, and I think they put us in LSD 309, which really isn't an ideal lab for this class, but we'll do our best. Okay. All right, guys. So I will see you next week. The lecture I will have posted on eCampus later today. I think that's about it. So I'll see you guys Tuesday. Remember, we'll continue with this in class. We will have lab next week in LSB 309 at 2. So if there's no questions, I'm going to end the Zoom meeting, and I'll see you guys next week.